Good afternoon. Embryology, megasporangium, and female gametophyte. Female gametophyte development. How to female gametophyte development? These are things are we can learn about to the today class. What is the male gametophyte? What is the male gametophyte and how to use for the male gametophyte and what type of male gametophyte is the present in this one. And here we can study the how we will learn about we will discuss about to the female gametophyte. What is the female gametophyte? How many female gametophytes are there? Or what is the structure of female gametophyte? These are things. First one, female structure of ovules, structure of ovule, types of the ovules, structure and types of ovule. Next one is the megasporangium, megaspore tetraoids. Hello. Ma, you didn't pay the fee? Okay, well, you can go and meet the controller of examination. Okay. Hmm. Now you are not there in the college. Okay. Female gametophyte structure. The female gametophyte structure of a typical embryo sac. Next one is the develops within the sporophytic tissues. Sporophytic tissues. The sexual organs of the flower, the male gametophyte, developing pollen grain composed of two sperm cells encased within a vegetative cell, develops within the anther, fertile part of the stamens. The female gametophyte embryo sac develop within the ovule, which is found within the female gametophyte. See, this is the male gametophyte, style, stigma, ovary. This is the female gametophyte. This is the ovary. This part is called thalamus. This is the style. This part is called the stigma. The female gametophyte embryo sac develop within the ovule. This style, this one is the embryo sac developed within the ovule, which is found within the ovary. Gynecium or pistil. Next one is the that is the female reproductive organ in a flower, and carpel is a unit of it. A carpel consists of a basal swollen portion is present. That swollen portion name is called bearing. Some plants, the ovary bearing one or more ovules. One or more ovules. A receptive stigma and often a small stalk-like style in between them. Okay, where the stigma, one or more ovules, a receptive stigma, and often a stalk-like structure is style is becoming between them. Ovules are enclosed by the ovary wall. The part of the carpellary tissue to which the ovules are attached is called placenta. Ovules are enclosed by the ovary wall. The part of the carpellary tissues to which is the ovules are attached is called placenta. And the distribution of ovule in the ovary is described as placentation. The ovules are also known as megasporangium. The megasporangium formation of the megaspores and female gametophyte. Okay. What is the structure of ovule? The megasporangium or ovule consists of nucellus and its protective port is present. Here, the ovules, the structure is the first layer name is called integuments. It is attached to the placenta on the interior wall of ovary by a stalk is called funicles. Both are there funicle. Only single one is there funicle. And the point of attachment of ovule to the funicles is called hilum. A mature ovule ready for fertilization consists of the nucellus. Consists of nucellus. Enveloped almost completely by one or two sheets. Completely one or two sheets. Uh, Completely one or two sheets. That is called the integument. Outer integument, inner ovule. Uh, Yeah, here the name of the mature here a mature a mature ovule ready for fertilization. The consists of nucellus enveloped almost completely by one or two sheath. That sheath name is called integuments, leaving a one small opening at the apical end. This opening name is called microphone. Okay? The both combination of the another one is the structure of the basal part. Thus, basal region of the ovule, where it is attached to the placenta. That placenta bifunicule is called chalaja. So the chalagal end. It opposite end is termed as micropyle end. The main passage for the entire the pollen grains into the pollen tube into the ovule. The nucellus female gametophyte is present also, known as embryo sac. That name is called the embryo sac. Nucellar tissues. That one is the nuclear tissue is the parenchyma tissues. The total made up of nucellus tissue, parenchyma, and represent the wall of megasporangium. The nucellus is mostly consumed by the developing embryo sac or endosperm. Each ovule has only one nucellus. That is called, that is a nucellus, meaning the reserve the food material. Next, the ovule, which is a single integument is called, that is the unitegument. Only one integument is there, that is a single one, unitegment. Sometimes two integuments also present, that is called the bitegmic. And sometimes without integument, someone is the integument is absent, that is called ategmic. The outer layer of the ovule to cover by the layers, that layer's name is called the integuments. These integuments, some families, only one integument is there, unitegmic. Some families, two integuments or two covers are there, that is the bitegmic. Some families, integument is totally absent, that is the ategmic. Next one, part of the part of the ovule. See, this is the ovule structure. This is the micropyle end. This is the chalagal part. And this one is the raphe. And this one is the nucellus. And these are the integuments. One integument, two integuments. This is the ovule, the antipolar cells, central or two polar nuclei, egg operators. This is a synriged vascular strand, hilum. This is the funicle. Here, the first part name is called, you can observe here, funicles. Funicles are funicle. Both funicles are funicle. A stalk-like structure which ovule is attached to the placenta. Where the ovule is there, the placenta is present. In that placenta, to attach the stalk-like structure, that is called the funicles. Nucellus. See, this one is a nucellus. The body of the ovule name is called nucellus. The total body of the name is called the nucellus. What are the integument? This is the covering, protecting layers, where the ovule protective covering of nucellus is called integument. Inside the ovule, the total body of the ovule This micropyle, small opening, two, two integuments over nucellus. Chalaja part. See, this one is the chalaja. The basal portion of the chalaja, the basal portion of the chalaja is called the ovule. And the basal part name is called chalaja, where the ovule. And hilum, this one is the hilum. A region where ovule fuse with the funicles. The ovules, all ovules fuse with the funicles, that is called the hilum. See, this one is the embryo sac. Female gametophyte located in the nucellus. Female gametophyte located in the nucellus developed from megaspore. Next one, types of, this is a part of ovule is complete. Next, types of ovule. On the basis of the position of the micropyle, the main position of the basis of the position of the micropyle with respect to the funicles. Mature ovule can be divided and can be classified into 6 types of ovules. One is the orthotropous, second one anatropous, third one campylotropous, fourth one amphitropous, fifth one hemianatropous, sixth one cercinotropous. Basic on the position of the micropyle, these are the 6 types of ovules. See, this is the first ovule is called orthotropous. See here, this is the structure of orthotropous. The main all ovules, the basis position of the micropyle, they can be differentiated are there. The first one is the orthotropous. This is the micropyle. These two are integument, outer integument, inner integument. This is the embryo sac. In this one is called the nucellus. And this part, basal part is the chalaja. And this part is called the funicle. Okay, here, orthotropous ovule. Another name is called, also known as called atrophous. Orthotropous or atrophous ovule. It is atrophous ovule. It is upright. Here micropyle is upright. Upright in this type of micropyle, chalaja and funicle. Both three. See, this is a micropyle. This one is the chalaja. This one funicle. We can observe both both on the one, both are all are the is one straight line. This one, this for example, this ovule is called Polygoniaceae and Piperaceae family. In this type of ovule, you can observe that one Polygoniaceae or Piperaceae is called, the name is called atrophous ovule, orthotrophous. Another name is called anatrophous. See here, you can observe the anatrophous. The micropyle totally bend, the opposite of the, and here you can observe the micropyle, funicles, hilum, both are same line. Where the upper part you can observe the chalazal is there. And this is the integuments, this is the raphe, and this one is one, this one, anatrophus. In this type, funicles is long. The body of the ovule becomes completely inverted so that micropyle becomes to lie close to the base of the funicle. This happened due to unilateral growth of the ovule. This is the structure to happen. How to happen? That is the unilateral growth of the ovule. The nucellus remains straight, so micropyle and chalazel lie in one line, and funicles lie parallel to it. It is the most common type of ovule that is called anatrophus, all angiosperms ovule. Coming to the third one. The third ovule name is called campylotropha. See, this is a, here you can observe, curve-like system are there. This is the micropyle, this is the chelagel, and this one is the funicle. Here, in Campylotropous ovule, body of the ovule is not completely inverted. The curvature is less than that in Anatropous ovules. The micropyle and chelagel do not like in the straight line. Here, the micropyle lies at the lies at right angle to the chelagel. In the example, Caparidaceae, Chenopodiaceae and Capariaceae families, you can observe to the Amphitrophus. See, this is the Amphitromus. 160 degrees curve is there. It is similar to the Campylotropous ovule. But in this case, the curvature of the ovule also affect the nucellar or embryo sac, so that it went like horseshoe like shape is there. This one is called the amphitropus. Another name is called the horseshoe shape ovule, as in Butamaceae, Alcinaceae family. See, you can observe this one. This is the integuments. This one is the embryo sac. That embryo sac is the horseshoe shape, and in this body is called nucellus, and this part is called chelaja, and this part is called the raphe, and these two is called the integuments. This is called the amphitropous ovule. Coming to the hemianotropous ovule. Hemianotropous ovule also, hemianotropous ovules are also known as hemitrophous. In this type of ovule, the functions to the functions funicles is not right angle integuments. Micropyle and chelagel lie in the same line in the Ranunculacea, Gnathosporidium, and Thulvegia. See, this one is a hemianatropous diagram. This is the hemianatropous. This is the picture of the hemianatropous. Here, this part is called micropyle. These three are called integuments. Inside the embryo sac is a liquid-like structure that is the nucellus, and this part is called the chilazal, and this one is called embryo sac. Where is the funicle? And this is the picture of the hemianatropous. Coming to the Cercinotrophus ovule, a very peculiar type of ovule is seen in the some members of the Plumbaginaceae, Plumbaginaceae are the family, are the nucellus protuberance is at first in the time. A very peculiar type of the ovule is seen in some members of Plumbaginaceae. Here, the nucellus protuberans is the first in the same line as the axis. But the rapid growth on one side causes it to become a anatrophus. Fuel has turned over completely so that they does not stop but continuous until the ovule has turned over again points upwards. It has been suggested that this kind of ovule also seen in Opuntia separatum name. This is called the Sarcinotrophis. See, this is the funicle, and this part is called the micro, and this one is the embryo sac, and here this part is called the chelazal part. These are all the six types of ovules. The first one is the parts of ovule. Next, different types of the ovules are complete. Parts of the ovule is called main parts of the ovule. Funicles are follicles. Finally, follicles are follicles. Second one is the nucellus. Third one, integuments are the provide covering of the nucellus. Next one is the micropyle, a small opening formed by two integument over nucellus. Next one, chalaja. Next one, chalajal, basal portion of the ovule. Next, sixth one is the hilum. Reason when we ovule which ovule is one anatrophus, third one campylotropus, fourth one amphitrophus, fifth one hemianatropus, sixth one is a sarcinotrophus. These are the female gametophyte ovules and types of ovules, parts of ovules. Coming to the development of female gamete. Megasporogenesis and types of megaspore. Development of megaspores, where it is known as megasporogenesis. Development of the megaspore within the ovule. Megasporangium is known as the megasporogenesis. A hypodermal cells in the nucellus, where you can observe in the hypodermal cell at the way the micropyle endodermal cells differentiated and function as the archisporium. Archisporium meaning that is a female gametophyte development cell. That cell name is called archisporial cells. During the initial stage of the development of ovule, primary demarked at the apex of the nucellus below the epidermis. It divides periclinical into an overprimary partial cell and partial cells are there. One is it divided into periclinical divisions. After periclinical divisions, to form two types of inner side primary sporogenous cells. The latter as a primary sporogenous cells, which is enlarged in size and divide mitotically after divisions, after after division of the micropyle, it divide into the pericytial and sporogenous. The later, the primary sporogenous cells, which is enlarged in size and divide mitotical division, form a row of four megaspora cells is total to form to the four cells. The upper cells degenerate while the lower part of the cell only function and it is called functional megaspore. This cells forms four haploid megaspores mitotically, which are arranged in linearly tetroid of the four megaspores. Three megaspores which are near the micropyle degenerate and one megaspore of the chelagel end become functional. This functional megaspore during development forms female gametophyte. The functional megaspore now form the female gametophyte that is called the embryo sac. Haploid tetroid of megaspore may be T-shaped tetroidal shape. T-shaped tetroidal arises due to the vertical division in the micropyle diet cells and transfer division in the chelagel division. See, this is a picture of embryo sac development. Primary archisporial cells. This one is the primary archisporial cells. These primary archisporial cells, primary pericytial cells, and after division, after polyclinical divisions, pericytial cells. Huh. See, this is the pericytial cells. Here, after divisions, pericytial cells, and the pericytial megaspore mother cells. Uh, and next one is the linear tetroid megaspore. Various stage. These one total four cells to form. In the four cells, three cells are degenerated. Only one is the development is there. See here, degenerating megaspore. Only how many? Only one is functional. That one is called the embryo sac development. Next part, female gametophyte. The structure and typical embryo sac. Here, the functional megaspore, the functional megaspore is the first cell of the female gametophyte. Next one, the cells enlarge and undergo. A functional megaspore is the first cell of the female gametophyte. The cells enlarges and undergoes three free nuclear mitotical divisions. Mitotical divisions. The first division produce two nucleate embryo sac. And the two nuclei shift to ends of the divide and thrice. Next one is here the first divisions produced. Okay, here the functional megaspore, the first cell of the female gametophyte. The cells enlarge and undergoes three free nuclear mitotic divisions. The first division produces two nucleate embryo sac. The two nuclei shift to the two ends and divided there twice, forming four nucleate and then eight nucleate structure. One nucleus from each side moves to the middle. They are called polar nuclei. The remaining three nuclei from cells at the two ends, three-celled operators at the micropyle end, and three antipodal cells at chelagal end. The two nuclei which migrate to the center called polar nuclei. The polar nuclei later fuse to form a single diploid secondary nucleus. This is a central. See, this is the development of embryo sac. Here, this one is primary archisporial cells. This wonder. This is the ovule primordium. This total ovules primordia, primary archisporial cells. After this one divided into one, this is the integuments. And the next one, primary pericytial, outer side, inner side, inner outer sporogenous tissue. After mitotical divisions, to form to the cells four haploid megaspores. This one is primary is always is there. This is not to under division. Only which one is division is there. Sporogenous cells is the undergo to mitotic division to form to the four haploid megaspores. That one, three are degenerate, only one cell to develop. This is the functional megaspore. See, this is three cells degenerated megaspore. This one is a, these three is the degenerate, only one is the functional megaspore. This is functional. This functional, this is the polar nuclei. This is the egg operators. Secondary nucleus. This one is the antipodal cells. These events result in a, see, surviving megaspore. See, these four cells nuclei is there. This three is degenerate. Only one is the developing is there. This part is the total ovule. And this is the integument. These three antipodal cell, polar nuclei, egg, synergid cells. The total, this is called the, to form to the embryo sac. These events result in a mature seven called structure called seven cell structure is called the female gametophyte or embryo sac consisting on the three antipodal cells. One central cell having two polar nuclei, two synergid cells, one antipodal cell, one central cell having two polar nuclei, two synergid cells, and one egg cell. Since this type of embryo sac developed from a single megaspore, this type of embryo sac to develop from the single megaspore and has 8 nuclei. It is said to be monosporic 8 nucleotype of embryo sac. This one name is called monosporic 8 nucleotype of embryo sac or polygonum type of the embryo sac. Types of embryo sac. Embryo sac is three different types of embryo sac is there. Monosporic embryo sac, bisporic embryo sac, tetrasporic embryo sac. The monosporic embryo sac, the polygonum type of embryo sac as described above in the formed from one of the four haploid megaspore nuclei, which in turn formed from diploid megaspore mother cell. As a result of meiosis, although it is the most common type of mode of embryo sac development in angiosperms, even there is a sustainable number of plants where more than one megaspore nuclei take part in the process. Therefore, depending on the basis of involvement of number of megaspore nuclei in its formation, the embryo sac can be differentiated type monosporic, bisporic, tetrasporic. Three different types of embryo sacs to development is there. That is called the monosporic embryo sac, tetrasporic embryo sac, uh, tetrasporic embryo sac. Monosporic embryo sac is one of the is the one where only one. This is the monosporic is the one of the is the one where only one of the four megaspore take part in the formation as the polygonum type. Thereof megaspores usually those which are now are at are at uh, micropyle end, degenerate, leaving only one functional megaspore. In this type of embryo sac, all the nuclei are generally published generally published genetically identical. This type of embryo sac nuclei are genetically identical because they are formed through mitosis of a single nuclei. The monosporic embryo sac embryo sac are further divided into two types. One is the polygonum type, another one is the Enothera type. Polygonum is called N8 nucleated. Enothera type is called 4 nucleated type. Next one is the polygonum or 8 nucleated type. Polygonum 8 nucleate as described earlier. It is formed by the chelazal megaspore of the tetroid and is 8 nucleated. A mature polygonum type of embryo sac compresses a three-celled egg apparatus, three antipodal cells. The total one is a mature polygonum type of embryo sac comprises and binucleated central cells. See, this one is the polygonal. This type of embryo sac is the most common and is therefore commonly designated as the normal type of embryo sac. However, it is also designed as the polygonum type because it was reported from the first time Polygonum divaricatum. Polygonum divaricatum. First one nucleus to divide two part. Again divisions, three cells and one cell to form. See again divisions. This is the 3, 3, 6, 7, 8 cell stage. That is the polygon. Coming to the second one, Enothera type. Here you can observe only 4 nucleate type. Enothera type of embryo sac is derived from the micropyle megaspore of the tetrad and is 4 nucleate. The mature embryo sac consists of an egg apparatus. Enothera type of embryo sac is found into the Anageraceae family. Anageraceae family. That's why in Enothera lamarckiana, the embryo sac is usually formed by the micropyle megaspore of the tetrad, which undergoes only 2 nuclear divisions instead of the usual 3 occurring in the polygonum type of embryo sac. In this way, 4 nuclei are produced which organize into the 2 synerged cells, the egg and single polar nuclei. Since the third division is omitted and all the nuclei are situated in the micropyle part of the developing embryo sac. There is neither a lower polar nucleus nor any antipodal cells. See, this is the Enothera type of first one to divide two cells, again two divisions in the four. See, this is the four cell type of the Enothera type. This is totally is the monosporic embryo sac. Uh, this one we will discuss about tomorrow bisporic embryo sac development. These are things we will discuss about to the tomorrow class.